Pawan, once you start the broadcast, I will check the YouTube and confirm.
Yeah, it is on right now, Basu. So,
okay, I'm just waiting for the feed.
Okay, I'm just waiting for the feed.
I think we'll get started in about a minute.
Yep, Pawan, we are good.
You're good? Okay, perfect. Yes, doctor, we'll start in a minute. I'll start off with the housekeeping and then we can get started. Yes, doctor, we'll start in a minute. I'll start off with the housekeeping and then we can get started. Okay. We'll get started. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನಾನು ಪವನ್. ಇವತ್ತಿನ ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಸ್ವಾಗತ. Today's event, Understanding COVID Vaccines, is the third in the series of COVID awareness programs that KSA is bringing to the community over the past year. Today's session, KSA brings you the session today in collaboration with the India Association of Phoenix and Jewel of the East Lions Club, Phoenix. We thank Mrs. Radhika Shiva, President of India Association of Phoenix, and Mr. Sridhar Chanagiri, IA board member, and the members of Lions Club for all their support in organizing this session. Before I introduce our esteemed speaker for tonight, a few reminders to our viewers. Given the nature of the topic for today and the wider audience and the community across the valley that we have today, the session will be in English. As you're aware, we are currently live streaming the Zoom meeting that we are currently on over our YouTube channel. And if you have any questions that you would like to pose to our speaker, please go ahead and post them on the comment section of the YouTube video that you are watching. Mr. Basavaraj and Mr. Shivakumar from KSA will be monitoring the comment section and will collate those questions and bring it up during the Q&A session as a part of this session. Without much further ado, let me introduce our esteemed speaker for tonight, Dr. Vikram. Dr. Vikram is a professor of medicine, Mayo Clinic College of Medicine, Vice Chair, Division of Infectious Diseases, Program Director, Infectious Diseases, Fellowship, and Associate Chair for Education, Department of Medicine at the Mayo Clinic here in Arizona. He is a fellow of the American College of Physicians and the Infectious Diseases Society of America. Dr. Vikram is originally from Bangalore, India. He earned his medical degree from Bangalore Medical College. He completed his internal medicine residency and chief medical residency at the University of Connecticut, followed by a postdoctoral fellowship in infectious diseases at the Yale University School of Medicine in New Haven, Connecticut. He joined Mayo Clinic in Arizona in 2005. Dr. Vikram was the 2019 recipient of the Distinguished Educator Award, Mayo Clinic's highest honor for sustained contribution towards education and leadership. In 2020, Dr. Vikram was one of the recipients of Mayo Clinic's Top Provider Recognition Award. He was recently named as one of the 2021 top doctors in infectious diseases in Phoenix Magazine. Dr. Vikram's primary areas of research include cardiovascular infections, organ transplantation, and immune-compromised host infections, and valley fever. Dr. Vikram and family are lifetime members of the Kannada Sangha of Arizona, and we thank them for that. He had previously conducted 2 COVID awareness sessions through KSA in March and May of 2020. We are indeed honored and privileged to have Dr. Vikram address the session tonight. And Dr. Vikram, thank you for your time today, and over to you.
Thank you so much, Mr. Pawan. I'm hoping that you can hear me well.
Yes, I can, Doctor. Thank you.
Thanks again to KSA and its entire leadership team, and for our other patrons who are joining us today, and also for giving me this opportunity for the 3rd time to come and address our audience on a topic related to COVID. I would also like to welcome each and every one of you who are on this session for giving up an hour or 2 of your time on a weekend evening and to join us to learn something about COVID together. This topic was particularly difficult to prepare for several reasons. One of them is the rapidity with which things are changing in the realm of COVID vaccines, as well as the
Pawan, you may want to mute your microphone.
Infectious diseases at the Yale University School of Medicine in New Haven, Connecticut.
Second.
In Arizona in 2005. Dr. Vikram was the 2019 recipient the distinct
Sorry about that. So anyways, the reasons why this has been such a challenge was because of the rapidly changing developments with COVID vaccine almost on a daily basis. And as most of the information is coming through media channels and news releases rather than publications, especially in real time. So what I have done today is to go over my screen. Let me just share my screens here. Perfect. I'm hoping you can see my slide.
Yes, Doctor.
Perfect. So what I'm planning to do today is to talk about the basic immunology of how vaccines work in humans, then talk about various forms of non-COVID vaccinations that are available and how they work. Let's talk about vaccine efficacy measures in studies when we look at vaccines and study them before they are released. Look at SARS-CoV-2 structure and targets for vaccines, types of COVID vaccines, and then a few minutes on emerging SARS-CoV-2 variants and how that impacts on our vaccine development. And then we'll go through question and answers beyond that. So if we look at cells that are present in our blood, we have several different varieties as is seen here. Ones that are listed inside the box are known as white blood cells. And these are mainly responsible for our immune system and for immunity against pathogens mainly. among white blood cells, there are various varieties as is listed here. When we talk about our immune response, there are two main types of immune responses. One is known as innate immunity, and the other is adaptive immunity. Innate immunity is something that our bodies have to respond when it comes across pathogens immediately, because there is no time for the adaptive immune response to react as yet, because adaptive immune responses take anywhere from 2 to 3 weeks to develop. So innate immunity is present in all vertebrates and all living beings for that matter. And the major innate immune responses include our skin, which provide physical barriers, the lining of our intestines and our lungs, which also provide a physical barrier. But beyond that, once pathogens gain entry beyond the physical barriers, then we have our neutrophils and macrophages, which try to engulf these bacteria or pathogens and then try to get rid of them. So that's the innate immune response, which has to happen right after pathogens gain entry. Beyond that, we have adaptive immune responses. And adaptive immune responses are those that our body learns after exposure to pathogens. And here, they also have memory, meaning that once a pathogen is seen and an immune response is developed, then if the same pathogen is exposed downstream, our adaptive immune response can recognize the pathogen. And the main cell type that is responsible for adaptive immune responses is the lymphocyte. So if you look at the lymphocyte, there are two main types of lymphocytes, T lymphocytes and B lymphocytes. And B lymphocytes are the ones that release antibodies, which bind to the pathogen, let's say viruses in this case, and then the viruses are rendered ineffective or they're consumed by other immune cells. T lymphocytes are of two varieties, helper and cytotoxic cells. Helper cells have two functions. One is they stimulate the B lymphocyte to release antibodies, and they also activate cytotoxic T lymphocytes, which are the ones that destroy infected cells. So pathogens, once they enter the body, if they are being assessed by the adaptive immune response, there are two things that happen. One is antibodies bind to the pathogen, and then cytotoxic cells will destroy the pathogen directly. So there is the antibody-mediated immunity, and there is cell-mediated immune responses. So let's take the example of a virus that gets inside the body. It multiplies within the cells, and then the virus replicates. And the viral antigens, which could be any part of the virus, is picked up by these immune cells. And these antigens are then processed by the helper cells. And as we said in the prior slide, the helper cell will then lead to both cell-mediated immunity, which is the cytotoxic cell, which goes after the cells that have virus inside them and destroys it, or the antibodies will be released by plasma cells, and antibodies will bind to these viruses, rendering them ineffective. Now, instead of natural immune response, now let's replace the active infection by the pathogen by a vaccine. So vaccine will introduce parts of the virus or viruses that are inactive, and the body then does the exact same thing, which is it recognizes the antigen, and then the immune response is stimulated. And in case you get exposed to the active infection in the future, you have preformed antibodies as well as cell-mediated immune responses that have been primed or triggered, which will then be ready to act on the pathogen once it's exposed. What are different types of vaccines that are available? So one variety is the live attenuated vaccines. Now we are talking about non-COVID vaccines. These are all approved vaccines. So live attenuated vaccines are those where the live pathogen has been rendered weaker by various methods, so they cannot replicate and they cannot cause disease in humans. So examples of live attenuated vaccines include your measles, mumps, and rubella vaccine, chickenpox vaccine, and some others. Live attenuated vaccines, for the most part, lead to long-lasting immune responses. The other variety is the killed vaccines, and killed vaccines are where the pathogen is administered inside the host. However, the pathogen is entirely killed, but the body reacts to the antigens that are present within the killed virus. And one good example is the killed poliovirus vaccine or the pertussis vaccine. Instead of an entire killed virus or pathogen, a part of the virus can be introduced. That's another vaccine, for example, some seasonal influenza vaccines. Then you can have virus-like particles that only have the viral shell, but not the genetic material, so it does not reproduce within the human host. One example is the human papillomavirus vaccine. Then you have toxoid vaccines, wherein certain pathogens like diphtheria and tetanus release a toxin which causes most of the damage. In this case, the toxin itself is inactivated and injected as the vaccine, and it elicits an immune response. You then have recombinant subunit vaccines, where a portion of the virus or bacteria is genetically produced in another pathogen, and only a portion of that is immunologically active is made into the vaccine and then injected into humans. And the first recombinant vaccine that was developed was the hepatitis B vaccine. Then you have conjugate vaccines, where a part of the virus or bacteria is taken. By itself, it cannot induce a long-lasting immune response, so it is conjugated to an adjuvant, and this can be a variety of products, and the adjuvant will boost the immune response to the part of the virus or bacteria and will lead to a very sustained and long-lasting protection. And this is another vaccine which does not have the conjugate. So these are some of the varieties of vaccines that we currently use in clinical practice, both in children and in adults. Now let's come to SARS-CoV-2, the virus that causes COVID-19. And this is a diagram that represents the structure of SARS-CoV-2. So as you are all aware, this is the virus, and this is the spike protein, and this is the cell. This is the cell membrane. These are proteins here, and this is the genetic material. This is the RNA of the virus. SARS-CoV-2 is an RNA virus. This is an enlarged version of the spike protein, and the reason why this is very important for today's discussion is because all of the vaccines that we have try to make antibodies against the spike protein of the virus. Especially, this is known as the receptor binding domain of the spike protein. So here's what happens. When the virus comes near the host cell, the virus has to bind to the host cell receptor, the host being us humans. The receptor in the host to which the virus binds is known as ACE2. And if this is the receptor, the spike protein, the S1 portion of the spike protein binds to this ACE2 receptor. That's the first thing that happens. So what happens next? Once the spike protein binds to the receptor, then there is a change that happens in the proximal portion of the spike protein. So this change leads to the rest of the spike protein to fuse with the human cell membrane. And this process then leads to the virus gaining entry inside the human cell. So receptor binding and then cell fusion. Once cell fusion occurs, then the virus gets inside the cell and then releases its RNA. And the messenger RNA is then translated into the genetic code is released, it's translated, and then the human cell will use that message from the virus, and then it will transcribe each of the proteins that are coded by the genetic message from the virus. So the genetic message is converted into proteins that are produced inside the cell. Each of these proteins are then assembled back to create a new viral particle, and the new viral particle will then exit the human cell as daughter cells. So not one, but many of these viral particles are produced, and they will then come out of the cell, destroy the cell in the process, and then each of these viral particles will infect other human cells, and the infection will escalate and continue. So now, what can we do to prevent this process? Studies have shown that the initial step, which is the spike protein that binds to the human cell, is the crucial step. And if we can avoid that step or prevent that step from occurring, then we can prevent viral infection. And therefore, if we can develop antibodies or proteins to this spike protein that attaches to the cell, then that can neutralize the virus completely, and the virus will be unable to enter the human cell, and it can be killed by our immune system. So as you can see here, these are the various proteins that the virus has, but the neutralizing antibody that can neutralize the virus is only against the spike protein. So using this terminology and using this concept, what our scientists have done is that they're working on various types of vaccines against SARS-CoV-2. So there are live attenuated viruses that are being studied. None of them have been approved as yet, similar to our measles, mumps, and rubella vaccine, where the entire virus is attenuated. There are inactivated viruses, which means that the virus is completely killed and inactivated. So this is the principle of the Indian Covaxin. Then you have a viral vector vaccine, which is a new type of vaccine wherein the genetic code for the spike protein is present within a viral vector, which is the adenovirus, which is the AstraZeneca vaccine, which in India is called Covishield, and the Johnson & Johnson vaccine. Both of them are adenovirus viral vector vaccines. Then there are subunit vaccines that are being developed, and subunit vaccines are where just the spike protein is produced, and then that is injected into the human cell. And Novavax is one of the subunit vaccines that's being studied. And then we have nucleic acid vaccines, and the two most common nucleic acid vaccines that are now being administered are the mRNA vaccines, which are the Pfizer vaccine and the Moderna vaccines. So what is supposed to happen here is this is the genome of the SARS-CoV-2 virus. And based on advances in science and genetics, within a few weeks after SARS-CoV-2 was identified, the entire genome was sequenced, and it was available for all of the scientists to study. And this is the genome that's present within the virus. And as you can see, if the brown portion is the genetic code for the spike protein that's present here, this is the genetic code that our scientists were interested in trying to develop a vaccine using that genetic code. So how do mRNA vaccines work? So what happened is that our scientists took the genetic code for the portion that synthesizes the spike protein, and they were able to sequence it and synthesize it. So what happens with mRNA vaccines is the spike protein mRNA sequence is produced in the laboratory, and that sequence is then placed inside a lipid nanoparticle because it's very unstable. And this is the mRNA vaccine, and this has to be frozen. The vaccine is then injected into the muscles into the arm of humans. And then the vaccine, what happens is this nanoparticle, which has the mRNA, is then taken up by local cells within the muscle itself. And then the genetic code is released into the human cell. So the human cell looks at the genetic code and only synthesizes the protein that is dictated by this genetic code, which is the spike protein. It does not synthesize any other proteins of the virus. And once the spike proteins are produced by the human cell, they are released from the human cell, which is your local cells within the muscle where the vaccine is injected. And once the spike protein comes out into your circulation, then the body's immune cells will start making antibodies against the spike protein, thinking that it's a live virus. And our B cells will make antibodies, and T cells will also be primed, and they will be ready to kill if the virus comes into contact with the human 2 to 4 weeks after the vaccine. Both arms of the immune system are ready to attack the virus. So this is how mRNA vaccines work. How about the adenovirus vector vaccines, which is the Johnson & Johnson vaccine and the Oxford AstraZeneca vaccine? Here, what happens is, once again, the genetic code or the mRNA that synthesizes the spike protein is removed from the virus, and a synthetic code is prepared in large quantities. And instead of that being within a lipid particle, like in the case of mRNA vaccines, here the mRNA is injected inside another virus. And this is known as the adenovirus. Adenoviruses are also common viruses that cause respiratory infections in humans. However, the virus that has been chosen to carry this genetic material from SARS-CoV-2 is either a human adenovirus 26 for the Johnson & Johnson vaccine, or a chimpanzee adenovirus, which does not infect humans, in the Oxford AstraZeneca vaccine. So both of these adenoviruses by themselves are replication incompetent, meaning that they are not live viruses. They cannot replicate once they're injected into the humans, and they themselves do not cause disease. They are just used as vehicles to deliver the spike protein genetic material into humans. So this is the vaccine. The vaccine is once again injected, and then the virus gets inside human cells and then just delivers the spike protein genetic material inside the human cells, and then the virus goes away. The human cell will process the genetic material similar to the mRNA vaccine, releases the spike protein, and this will lead to an immune response. So this is how adenovirus vector vaccines work. I hope you're with me so far. Another realization for all of us is the rapidity with which vaccines were developed. This was absolutely unprecedented. If you look at the top diagram here, for traditional vaccine development, it takes about 15 years or longer. First, you have to do animal studies. It should be safe in animals. Then you submit the investigational new drug protocol. Then you go through phase 1, phase 2, and phase 3 trials, which will take several years. Then you submit another application to the reviewing committee or board, and then they have to review the data and approve the vaccine. And then there is large-scale production, which takes about 15 years. What happened with SARS-CoV-2 vaccine development? So the same process. However, there was an overlap of phase 1, phase 2, and phase 3 studies, which were happening to some degree simultaneously, as they found that phase 1, the vaccine was safe, the vaccine was effective. Then they started studying the vaccine in humans in large numbers, and they submitted the authorization. The regulatory bodies quickly reviewed the studies that were available, and within 10 months after the vaccine development started, and this is especially because of all of the efforts put in both by industry and by scientists, we had a vaccine that was ready. This is an important slide for you to understand when you're looking at vaccines. And why this is important is because how do we define vaccine efficacy? Most of the studies, either the mRNA vaccines or the adenovirus vector vaccines, If you look at vaccine efficacy, the primary endpoint is to prevent infection in the people who received the vaccine compared to those who did not receive placebo. However, the other efficacy points are harder to study, but they are probably more important. For example, if you get a vaccine, even if you get infected, let's say, it causes minimal disease, that's probably not as bad as if the vaccine can prevent symptomatic disease, it can prevent hospital admission, it can prevent severe disease, such as being admitted to the ICU, or prevent death. So each of these are different time points or different vaccine efficacy endpoints that we will have to look into carefully when we address each of the vaccines. So if one vaccine does not have excellent efficacy at preventing infection, but still is able to prevent hospital admission, prevent intensive care unit admission or death, the vaccine is still effective. So let us compare the three vaccines that are currently available in the United States head to head. Pfizer is in green, Moderna is in beige, and Johnson & Johnson is in pink here. So if you look at efficacy based on age, sex, race, ethnicity, baseline body mass, and pre-existing conditions in the patients who were studied in these studies, they had similar vaccine efficacy and overall efficacy pretty much for all three vaccines. Dosing for Pfizer is 2 doses, 21 days apart. Moderna is 2 doses, 28 days apart, and J&J is a single dose vaccine. mRNA and mRNA vaccines here, this is a viral vector as we talked about. Authorized ages so far, Pfizer is 16 and older, Moderna is 18 and older, and J&J is also 18 and older. How many people were studied in their phase 3 clinical trials? For Pfizer, it was about 43,000 patients, half and half who got the vaccine versus placebo. Moderna was about 30,000, and J&J had about 43,000 patients. So large scale trials. Now, this is the slide that's important, just as we talked about vaccine efficacy. So what I would like to bring to your attention here is if you look at each of the three vaccines and look at their efficacy to prevent infection 28 days after, and prevent severe infection 28 days after vaccination, prevent hospital or ICU admission or death after vaccination, it is close to 85 to 100% for all three of the vaccines. These are hard and critical endpoints. If you look at preventing infection overall, that means that once you've had the vaccine, you get tested with a nasal swab to see if you get vaccinated, if you get infected in the placebo group or the vaccine group over the next several weeks after the vaccine. So just with prevention of infection, J&J appeared to be 72% effective compared to 94 or 95% for these vaccines. And the Oxford AstraZeneca vaccine had an efficacy for this particular outcome in the 70% range. However, these other endpoints were very similar. And effectiveness over age 65 was also excellent. Contraindications, pretty much all of the vaccines have similar contraindications, which is severe allergy to any vaccine ingredient or prior doses of the same vaccine. And you need to exercise caution if you have a history of severe allergy to any vaccine or injectable medication. And we will talk more about that during the question and answer session. Are they effective against variant strains of the virus? Not studied in large numbers as yet. Those are ongoing. J&J had some efficacy over these strains because the studies were conducted during the time that these variants were available or they were in the community. And we'll go over these variants in a minute. And can the vaccine cause infection in humans? Answer is no to all three vaccines. What about side effects? As you can see here, 60, 80, 50%. So most of the patients may have some side effect or the other, and side effects are especially seen after the second dose of the vaccine, especially in people who are younger, less than 55 years of age. And the most common side effects are injection site pain, headache, fatigue, and body aches. And sometimes you can get swelling of your lymph nodes in your armpits, which is adjacent to where the vaccine is administered. And some patients can have anaphylaxis, which is an acute allergic reaction. We'll talk about that later. Well, all of the industry-sponsored phase 3 trials showed efficacy of the vaccine, but people can remain skeptical, saying that do these vaccines really work once they are released into the community and millions of doses are administered to large populations? So what happens then? The best evidence we have so far that it works is the nationwide mass vaccination program by one of the major health networks in Israel. And they used the Pfizer mRNA vaccine, and this was published earlier this month. So they looked at 600,000 people who received Pfizer vaccine, compared them to 600,000 who did not get the vaccine. And look at the numbers here compared to 30,000 or 40,000 people during phase 3 trials. So vaccine efficacy, which is 7 days after the second dose of the vaccine, look at the numbers once again. 92% prevention of infection, 94% prevention of symptomatic COVID, 87% reduction in hospital admissions, and 92% prevention of severe disease. And these findings were essentially consistent with the randomized trial. So this certainly gives us some hope and encouragement that these vaccines do work in the population setting. And this is just graphs showing the same thing for various indications. So now talking about the 2 vaccines that are available in India. The 2 vaccines that are being administered, there are several others that are currently being studied throughout the world, but the 2 that are being administered, one is Covaxin and the second one is Covishield. Covaxin is the inactivated or killed whole virus vaccine that's made by Bharat Biotech. And Covishield is the same vaccine as the Oxford AstraZeneca vaccine, which is an adenovirus, chimpanzee adenovirus vector vaccine that's being administered. Both are administered as 2 doses given either 3 to 4 weeks apart. Now, after all the encouraging news, some cause for concern. So what has happened is there are several mutants or variants of SARS-CoV-2 that have emerged. Is this something that's new? Answer is no. Variants and mutations in respiratory viruses, especially with any RNA virus, is very common. It happens all the time. However, there are some mutations in these viruses that are particularly concerning because some of them have increased the transmissibility of the virus. They've made the virus more transmissible, and other mutations have caused them to have more severe disease in humans. And other mutations have led to reduction in neutralization by existing vaccines and antibody therapies, such as monoclonal antibodies. So those viruses that have these types of mutations have been labeled as variants of concern by the CDC, and they're being closely monitored. So let's look at a few of them that are of importance to us. So the first one is the B117 variant, which is the one that was first described in the United Kingdom, started there sometime in the fall of 2020, and then it is now the predominant strain of SARS-CoV-2 in the UK. So what does this variant do? It is about 50% more transmissible than the previous strain of coronavirus. It has increased severity of disease. This has been well shown in studies that have since been published after December. However, good news is for the B117 UK variant, the current vaccines that we have, all of the current vaccines work very well. So there is no impediment to vaccine efficacy. And we will talk about what's happening to this strain in the United States in one of the next slides. The more concerning variant is the B1351, which is now the predominant strain in South Africa. So this variant is a notch above the UK variant because this has another mutation known as the 484K, which makes this virus have mutations, especially in the receptor binding domain of the spike protein. So this is the area where the antibodies that are generated by all of the current vaccines go and bind to inactivate the virus. Therefore, this particular variant, when they have studied vaccines against in populations where B1351 is prominent, the vaccine studies have shown that consistently this variant can trump the vaccines, or in other words, vaccine efficacy is decreased in countries and populations that have B1351 variants. Likewise, there is moderate reduction in neutralization by convalescent plasma and monoclonal antibodies. A very similar variant with mutations is also known as P1, and this is mainly seen in Brazil at this time, where there are large outbreaks of this virus, which has taken over as the predominant strain. And P1 is similar to the 351 strain in South Africa in that it is more infectious, number one, and number two, antibodies produced by current vaccines don't bind to this virus very well and cannot neutralize the virus as well as the previous strains. And therefore, there have been breakthrough infections with this variant, even in people who have had infections before with the other strain or who have had the current vaccines. Something that's closer to us than you would want to believe. These are two variants, 427 and 429, that were first described in California. Both of these strains also have mutations in their spike protein, which leads to impact on neutralization by some of the monoclonal antibodies, and there is moderate reduction in neutralization by the current antibodies given by any of the vaccines that we have. So the current vaccines, their ability to neutralize this virus is not completely blocked, but it is reduced compared to the previous strain. So B427 and 429 is now the predominant strain in California. So if you look at this is an important slide. I just picked this up from the CDC website because it changes every day, literally yesterday. So this is what has happened to various strains in the United States, because we are now genotyping thousands of strains as the CDC gets hold of them and the state health departments get strains of coronavirus. They are sequenced to try to find which mutations they possess. So this is the 2-week period in January of 2021 to a 2-week period in March of 2021. So look at all of these variants that are going up in the United States. So B117, which is the British variant, is trying to become the predominant strain, and it will happen within the coming months. 1429 is the California variant, so that's going up in the United States, and others are also creeping up. Both the South African variant and the Brazilian variant have also shown up in several cities in the United States, and the exact level of transmission is being studied. So here is another alarming statistic for you, which is in California, the 427, 429 variant was first seen, and it's the predominant strain. 52% of their strains are the 427 or 429. Which are the other two states which have a lot of this virus? Adjoining states, Arizona and Nevada. So Arizona, 25% of the strains belong to the California variant, and in Nevada, 41% belong to the California variant. Look at all of the other states, they are less than 5%. So this is another cause for concern, and also another plea to the local public to get vaccinated soon so that these variant strains don't come into our communities and establish residence. Now, I mentioned the word monoclonal antibodies a few times. So what are these? Monoclonal antibodies are synthetically prepared antibodies against the spike protein. So what happens here is, as I mentioned earlier, once you get infected with COVID, it takes at least 2 weeks for our body to generate antibodies to protect you from either getting the infection again or to make the infection go away. However, during the initial phases, as soon as you're exposed to the virus, if we can inject antibodies directly, synthetic antibodies, then perhaps we can block the viruses and prevent them from infecting more cells, and thereby hope to prevent severe disease and hospitalization. So these are the monoclonal antibodies. There are 2 of them in combination. And this has been a very significant development in the last 3 to 4 months, which is in people who are at high risk for severe COVID-19. Once they get infected with COVID, if they are identified and if they receive the monoclonal antibody infusion within the first week after infection, then it has been clearly shown that the monoclonal antibodies can prevent severe disease and death in this vulnerable population. And we have administered this to hundreds of patients in the last few months. And the key here is to identify the high-risk patient right after they get infected and within the window when the monoclonal antibodies work. And the reason for me putting up the slide here is because some of these monoclonal antibody combinations, when they have been tested against the new variant strains, they actually are unable to neutralize the virus as well as they can with the previous strains. So this is another reason to get the vaccines sooner than later to prevent establishment of variant strains. So what I will do now is I will stop sharing my screens and take a sip of water and see if we have some questions from one of our volunteers who have joined us here.
Thanks, Dr. Vikram. So we're starting the Q&A sessions now, and we do have a youth representative from the Phoenix Jewel of East Lions Club with some questions. Adarsh, please go ahead.
Thank you. Good evening, everyone. My name is Adarsh, and I'm a 7th grader at Scottsdale Preparatory Academy. Tonight, I am representing Phoenix Jewel of the East Lions Club. Thank you to KSA for providing us with this opportunity. Phoenix Jewel of the East Lions Club is a volunteering organization that has been very active throughout the COVID pandemic. We have done many charitable and humanitarian services, such as feeding the homeless, helping in the resettlement of refugees, and volunteering at Bethany Home Pharmacy to aid in the efforts to provide COVID vaccinations, among many other projects. I now have a couple questions regarding the COVID vaccine in children for Dr. Vikram. First of all, are COVID vaccines effective in kids younger than 16 years? When will they be available?
Oh, thank you for the question, Adarsh. So let me share this one slide here. So that's a great question. Now, all of the 3 vaccines that have been approved in the United States were studied in those over the age of 16 or 18. And therefore, we don't have actual data in children. However, as you can see here on the slide, Pfizer has completed a study in children between the ages of 12 and 15, and their study results are awaited. Moderna has an ongoing study in kids 12 to 17, and it has started a study in children between the ages of 6 months and 11 years, and this study's results are awaited. And Johnson & Johnson will also begin studies in children. Give me just a second here, sorry. So at this point, when will the vaccines be available for children under the age of 16? There are some reasons why vaccines haven't been approved as yet. One is that this vaccine has not been studied. And secondly, in young kids, there has been a rare complication of COVID known as multi-system inflammatory syndrome in children. This is extremely uncommon. Between May of 2020 and March of 2021, there have only been about 2,600 cases that have been reported. However, this is a severe manifestation of COVID-19, and kids are being hospitalized with multi-system organ involvement. So the concern had been in the past that younger the person, the more likely that they will have a more significant reaction because of a robust immune system. So that's being looked into. And as soon as results are available from many of these trials, I would think that the vaccines should be approved for children who are younger than 16 years of age. So that is going to be happening soon.
Okay, thank you so much for that. And my second question is it safe for kids less than 16 to attend in-person classes, sports, and gatherings before receiving the vaccine?
Yes, it's a tough question to answer, and the short answer is yes. Kids can go to in-person schools, and that is based on data. In fact, there were 2 papers that were published in the CDC's journal, MMWR, just earlier this week. And one of them was from elementary schools in Salt Lake City, where they did not even have 6 feet of distancing for the entire time the kids were in school. And there was a high incidence of community transmission during this time. So even when the transmission was very high in the community, and they had 51 index cases of either kids or teachers who were coming to schools, they tested 1,000 contacts of these kids, either people who were working in the school or other kids. And guess what? There were only 12 cases that were positive among all of the contacts among these faculty or students who were positive for COVID. And among the contacts, only 5 of these contacts who were positive were found to have school-associated transmission. What it tells you is the incidence of COVID in schools mirrors the incidence in the community. The transmission rates within the school itself is actually very small, as long as the schools practice social distancing and wearing masks. And if we can reduce the incidence in the community, the school incidence will go down even further. And therefore, one of the first things we can do is, if we can vaccinate the community as soon as possible, that will be more reassuring for children who are now doing online classes to go back to in-person classes as soon as possible.
Okay, thank you so much.
Thank you. So at this time, what I would like to do is go through a series of questions. These are based on questions that were submitted by KSA when they did their survey from our patrons, and also questions that we frequently get in the hospital from our patients and colleagues. So I will go through a few of these questions and try to provide my answers. And I will also, in the meantime, pause in between and see if Mr. Pawan has any other specific questions from the audience. So we'll go through a lot of questions in the next 30 minutes or so. Now, the first question is, which vaccine is better? If you have a choice, should you get one versus the other? This is a frequently asked question, and I would say that right now, any of the vaccines, if you can get it, please go ahead and get it. The small differences in efficacy were only with infection. They were not with hospitalization, severe illness, or death. All of the vaccines do a fantastic job. So any of the vaccines should be fine. Next question. How long does vaccine immunity last if you get 2 doses of the vaccine? We know at the present time that immunity lasts for at least 3 months, perhaps much longer. That's because that's how long some of the study participants have been looked at by antibody studies in their serum. However, the longer time frame that elapses between when the vaccines were released, we will have more data on how long robust immunity will remain, and we will be protected from the vaccine. So that information is yet to come. Next question is, will we need a booster dose of the vaccine? The answer to that is we are not sure as yet. Once again, we have to determine 2 things. One is, how long will antibody levels persist after 2 doses or 1 dose of the J&J vaccine? And number 2, how quickly will the virus mutate, and will the variant strains that can bypass the vaccine, if they become the predominant strain in the community, then we may have to get a booster with a vaccine that will be effective against these variant strains. The companies are already in the process of manufacturing newer forms of the vaccine that are effective against the Brazilian and the South African strains, but that will take some time. So once again, the sooner we can block this sequence by taking vaccines that are currently available, we can prevent other viruses from getting into our communities and becoming the predominant strain. Will COVID vaccines integrate into my DNA and change my genetic code? The answer is no, it doesn't, because the mRNA vaccines are not DNA vaccines. The mRNA, as I showed you in one of the diagrams, gets inside the cell and stays within the cytoplasm of the cell and does not enter the nucleus of the human cell. And once the mRNA goes into the cell, its half-life after injection is only about 8 to 10 hours. So the mRNA goes inside the cell, it releases its genetic code, the cell picks up the genetic code, then manufactures the spike protein, and the expressed spike protein can persist for several days and will stimulate our immune system, both T cells and B lymphocytes. And therefore, COVID vaccines that are currently available will not integrate into our DNA and change our genetic code. Next question is about serious allergies to the COVID vaccine. Now, what do we do with an allergic reaction? Allergic reactions can be considered severe if the person needs to be treated with an EpiPen, or if they need to go to the hospital. And serious allergic reactions are sometimes also referred to as anaphylaxis, which is an acute reaction where you develop hives or welts on your skin. You may have trouble breathing. You can pass out. And these usually happen within the first 15 to 30 minutes after you get either a vaccine or a medication or anything else you may be allergic to. And then you have immediate allergic reactions, which can happen within about 4 hours after getting the vaccine. And then there are other minor reactions, which are side effects, which are not allergic reactions. There was a paper that was just published that looked at both Pfizer and Moderna vaccines after it was released. So almost 10 million doses of Pfizer and about 7.5 million doses of Moderna vaccine. And this was through the VAERS system that's through the CDC, which is the Vaccine Adverse Event Monitoring System or reporting system. So what they found among 10 million cases of Pfizer, 10 million doses of Pfizer and 7.5 million doses of Moderna vaccine is that there were 47 cases of anaphylaxis with Pfizer and 19 cases with Moderna. And this gives us a rate of 4.7 cases of anaphylaxis per million doses for Pfizer and 2.5 cases per million doses of Moderna vaccine. And therefore, this reaction is extremely uncommon. And what they also found was the allergies were more likely to happen in people who have a history of having a serious allergic reaction either to other vaccines or to other medications or substances. And therefore, in patients who have allergies to other types of vaccines, it's important that you talk to your physician to see if any of the COVID vaccines are safe for you. If you have allergies that are not related to vaccines, for example, you have food, pet, environmental allergies, or latex allergies, then such patients might react to one of the vaccines. It's unknown. They should still get the vaccine, but they should be observed for 30 minutes instead of the standard 15 minutes after the vaccine. So let me pause there for a second. And Mr. Pavan, do we have any questions from the audience that I can answer? And then I'll go back to my list of question and answers. Thank you.
Yes, doctor. So we have a couple of questions that I would like to pose. In fact, we have a few. The first one being, right now, only the Pfizer vaccine is advisable for 16 years and older. But the question that we have is it even advisable for people between 16 and 19 to get a vaccine?
I'm sorry, I had a bit of audio delay there. Can you please repeat the question for me?
Sure. The question here is it advisable to get vaccine for 16 to 19 age group?
Great, thank you. Answer is yes, because the vaccines were studied in this age group for both Pfizer and Moderna vaccines. So if your child is between the ages of 16 and 18, then the Pfizer vaccine is advised. If they're 18 and above, then Moderna vaccine or the J&J vaccine can be administered. And they are essentially considered adults since they were included in the study. Even in that proportion of people who were in that age group, there were no additional complications or side effects in the 16 or 18-year age group. So answer is yes, they can get the vaccine.
Thanks, doctor. The question around the California strain and the South African strain, is there any efficacy data on the current vaccines on these particular strains?
We have some incoming data on these vaccines. As you know, the one vaccine that has been studied extensively in South Africa is the AstraZeneca Oxford vaccine, because a portion of their clinical trials were conducted in South Africa. Their efficacy in South Africa, it depends on which period the study was conducted, because the South African variant emerged towards the later part of 2020. So in patients who had the South African variant, vaccine efficacy with the AstraZeneca vaccine was much lower than what they had expected with the previous strain of the virus. Likewise, a similar reduced effect of the vaccines has been noticed with both Pfizer and the Moderna mRNA vaccines in patients who've had who live in an area where the South African variant is predominant. Also, when you study these viruses in the laboratory, for example, if you take the blood from patients who have received any of the current vaccines and you incubate them with the Brazilian virus or with the South African virus, then the amount of neutralization that's provided by the current vaccines against both of these strains is diminished, in some cases significantly diminished. However, it does not mean that the vaccines don't work, because what we cannot study in the laboratory is the cell-mediated immune system. We can only take their antibodies and see if the antibody neutralization works or not, and to what extent it's suppressed. But what happens inside the body, because we have both arms of the immune system working against the virus. So there is some degree of protection that's provided by all of the vaccines. It may not be as good as what it is for other strains of the virus.
Thank you, Doctor. That's it's very informative. This question is probably going around in everybody's head right now, and so the question is: there have been reports of many European countries temporarily holding AstraZeneca, but it is still being administered in India. Are there any risks to our aged parents and relatives back home taking this vaccine?
Great question. That was actually the next question that I had on my list of questions too. Let me just get one of these papers here just to give you some numbers. So let's talk about some rare and infrequent side effects, which I'm sure our audience is keen on hearing. Before we talk about rare side effects, there are a few things that I have to set the stage for. One is, what is the incidence of the uncommon complication or side effect or the disease process in the general population? What's the background rate in the community of something like this happening without the vaccine? Or what was the rate before the vaccines were administered? So that's one thing to know. And secondly, it's also important to remember that rare side effects may not be manifest during phase 3 trials. Looking back to what we just talked about, phase 3 trials had anywhere from 25 to 40,000 patients. If a very rare or uncommon side effect is 1, 2, or 3 per million doses of vaccine, that may not show up during the study period. So what is very important for all vaccines or any new drug that is approved based on trials is that there needs to be continued monitoring to see if any of the side effects will manifest once the vaccine is given to millions of patients and millions of doses are administered. So that's the key. Now, let's look at facial palsy. Facial palsy is paralysis of one side of the face. And if you recall, during the Pfizer vaccine trial, there were a few cases of facial palsy that were seen in those who got the vaccine and also in those who got the placebo. But when you look back at the rate of facial palsy that happens in the general population as a whole, whether you take the UK or the United States or any other country, the background rate of facial palsy in the community was actually slightly higher than the rate of facial palsy that was seen in either the vaccine recipient or in those who got placebo. So what does that tell you? It doesn't completely exclude the possibility that the vaccine may have caused facial palsy, but it gives us reassurance that it probably just that based on the community rate, that person would have developed facial palsy even if he or she was not in the study. The fact that they were in the study, it was a coincidence that they got facial palsy. They were being monitored, and we have to be careful to make sure that it was not due to the vaccine. So that's one thing to remember. So that's about facial palsy. So that's been carefully reviewed, and it's been determined that that was not caused by the vaccine. Now let's look at the issue of blood clots, which was in the news, and several countries in the European Union stopped using the Oxford AstraZeneca vaccine. So what happened was there were two different types of blood clotting events that occurred. One was the blood clots that were seen in the extremities and different parts of the body. And then there was another very rare form of blood clotting that happened within the skull, or within the veins, large veins surrounding our brain. This was called as cerebral venous sinus thrombosis. And that's a more serious complication, or an event, whether it happens because of a medication, whether it happens in the community or for various reasons. And none of these are just due to the vaccine, by the way. There are other reasons why some of these things can occur. So when Europe took a pause after it was reported that some people who received the vaccine within a few weeks after they had blood clots, then they looked at they went through the same exercise. So then they went through the same rates. So what they found was about 20 million people had received the vaccine. They had 7 cases of blood clots in multiple blood vessels. And there were 18 cases of cerebral venous sinus thrombosis. So they determined that the background rate of developing blood clots in the general population, and the older the person is and more comorbid conditions the person has, the chances of having blood clots increases. So it's quite common, and it's not an uncommon illness that physicians see both in the inpatient and outpatient setting when people have blood clots. So the rate of blood clots in general was not higher than the community. However, the number of cases of cerebral venous sinus thrombosis was slightly higher than what was expected in the general population. And this complication was also seen in younger patients in comparison, and more so in women. So they are looking into these cases in more detail, because if these patients had other reasons to develop these clots, or whether they had other predispositions, then it may not be related to the vaccine. So what the European agency has said is that at the present time in the news release, which was on the 18th of 2021, they indicated that the benefits of vaccine continues to outweigh the risks of side effects. The vaccine is not associated with an increase in the overall risk of blood clots. There is no evidence of specific batches of vaccines that cause this problem. However, the vaccine may be associated with very rare cases of blood clots associated with low levels of platelets, including the clots in the brain, that they're looking into more carefully. So the answer to your question after this detailed explanation is this has not been seen in their phase 3 trials in either in the United States or in other countries when the AstraZeneca vaccine was studied. These have still been isolated cases that have been reported in Europe. So more is being done. However, it is still an extremely uncommon manifestation, and therefore the benefits of getting the vaccine will definitely still outweigh any possible risks.
Thanks a lot, Doctor. That really helps. We have a few other questions coming in.
What I can do, Mr. Basav, let me take a few more questions that I have, and then I will circle back to you.
Okay, thank you.
So the next question that I have is, what should I do if I have side effects after getting any of the vaccines? And this is an important question, because we've seen some unusual coincidences which patients have attributed to the COVID vaccine. And let me give you some examples. If you develop pain in your arm, if you have some fevers and chills a day or two after the vaccine, that is likely due to the vaccine, especially after the second dose. You take Tylenol, you rest, and it goes away. It should not cause anything else beyond that. However, if you have symptoms that are unusual, or if you have symptoms that persist beyond the first 1 to 2 days, then you have to seek medical attention, because it may not necessarily be the vaccine. It could be something else that happened around the same time as you got the vaccine. So here are 3 examples. One is a patient who developed fevers and chills and lost a sense of smell and taste 3 days after getting the vaccine. He thought it was the vaccine. And when he came and got tested, he had COVID. So what happened here? What happened here is that the patient got exposed to someone with COVID 2 or 3 days before the vaccination. It takes about 3 to 5 days to develop symptoms, went and got the vaccine, and right after the vaccine had symptoms of COVID, not side effects from the vaccine. So that happens. So if symptoms persist, go get tested to make sure that you've not been exposed to COVID. Second example, we had a patient who developed high fevers, chills, night sweats, had a persistent cough, and then developed a whole body rash about 5 days after getting the COVID vaccine. She felt it was all due to the vaccine. Symptoms continued to progress, and then was referred to see me, and we tested her for valley fever, and she had acute valley fever. So once again, coincidence, she got exposed to the valley fever fungus coccidioides a few days or a week before the vaccine, asymptomatic, gets the vaccine, and then comes down with manifestations of valley fever. The third example is more serious. This was a patient, it was a kidney transplant patient who went and got his vaccine. 3 days later, developed high fevers, aches and pains, wasn't feeling well, thought it was all the vaccine, waited 3 more days, and then came to the hospital because fevers did not go away. And when we cultured his blood and did other tests, his transplant kidney was infected with a bacteria. And if he had stayed home for a few more days, things wouldn't have turned out so well. So the essence of this is minor side effects, let it go. If it resolves, let it go. If you continue to have symptoms, report it and seek medical attention. So next question. What about vaccination in those who have underlying medical conditions? Such as, let's say if someone's immune compromised, they take high doses of steroids, they have had an organ transplant, they have had previous cancer, they have recently received chemotherapy, they have an autoimmune disease. What about this group of patients? Each group is different, but as a broad category, can these patients get the vaccine? And the answer is an overwhelming yes. Vaccine can be taken by any of these groups of people. However, the extent of protection that's provided by the vaccine may not be as high as in someone who is not immune compromised, meaning their antibody levels may be lower. However, it's not zero. It depends on the type of immune suppression and extent of immune suppression. Since COVID, none of the COVID vaccines are live virus vaccines. It can be safely administered to those who are immune compromised, and we are doing that for all of our immune suppressed patients. It is especially important if you know that you're going to be immune suppressed, or if you're getting your next round of chemotherapy, to perhaps get the vaccine a few weeks before that, so that your body would have mounted an immune response when your immune system is closer to normal. And then even if you get immune suppressed, the the immunity from the vaccine would still be in your system. Next question, interpreting COVID testing in vaccinated patients versus prior infection. So if someone gets the vaccine, another common question that we get is, if I get my nasal test done, let's say before going overseas or traveling, will my test come back positive if I get the vaccine? Answer is no, because the vaccine only makes antibodies. It does not make other proteins, and it does not cause a nasal test to become positive. So if you have a positive nasal test, that means that you have COVID infection. Do we need to get an antibody test done to confirm vaccine efficacy? Answer is no, you don't. All of these vaccines, as long as your immune system is normal, they generate very high levels of antibodies in your blood, and therefore there is no need to check an antibody level. The only times we have done antibody levels to determine if the vaccine has worked is in people who are heavily immune compromised, to see if they have high levels of antibodies. If they have no antibody at all after the vaccine, at least it will help us counsel the patient to try to continue to maintain social distancing and masking, that they will likely remain at risk for getting COVID. So I can continue to move on because some of these questions might be what you may have in the chat box, Mr. Pawan. So the next question that a number of our patrons had asked is about pregnancy and breastfeeding. Is the vaccine safe during pregnancy or lactation? So, and that's a very personal question, and there is no clear answer. So here's what we know so far. Number one, none of the vaccine trials included pregnant or lactating women. And that has been the standard. Several of the associations have been trying to fight that or try to get them included in the studies. But when a new product is being developed, there are some populations that are excluded, and pregnant and lactating women are one group. So what is the data that we have about the vaccine during pregnancy? Number one is that animal studies, which are the phase 1 studies or pre-phase 1 studies, where the vaccine has to be studied in various animals, including pregnant animals, to see if there is any effect on either the mother or the embryo or the infant animal. None of those studies for any of the vaccines have showed any congenital abnormalities or any effects on the pregnant animal. So that's certainly comforting. Second is there are several other non-live virus vaccines that are routinely administered during pregnancy. For example, tetanus booster, pertussis vaccine, influenza vaccines, all of these have been administered to pregnant women over the years, and they are all approved. The Johnson & Johnson vaccine used the same adenovirus vector in a large-scale Ebola vaccine trial. And in this trial, the vaccine was administered to pregnant women in all trimesters, and they did not see any adverse pregnancy outcomes, either on the mother or on the infant. So that's also a good sign. Therefore, what should we do if someone's pregnant or someone's planning to get pregnant? So you have to look at benefits versus risks, right? So what are the benefits of getting COVID vaccine? So one thing we know for sure in the last year, after looking at patients, pregnant women who have developed COVID, especially during the later part of pregnancy, is that pregnant women with COVID, especially with severe COVID, have bad outcomes. One is they more often get severe disease. If they have severe disease, they more likely end up in the hospital or in the ICU, or they can die from it. And there is a much higher chance of a premature termination of pregnancy because of severe COVID disease. So during pregnancy, if someone gets COVID, the outcomes may be bad. So that's one. Second is, because of the above reasons, vaccine should more than likely be safe during pregnancy. Now, the other aspect to remember is, what's the risk of the pregnant woman? Does the pregnant woman have other risks beyond just being pregnant for COVID? For example, is she older, greater than age 35? Is she obese? Does she have other medical conditions such as diabetes or high blood pressure? Or if she's immune suppressed, that adds another level of risk for having bad outcomes during pregnancy. So, or if that person is a healthcare worker, if there is a higher risk of exposure as a result. So if the pregnant woman belongs to any of those categories, then the risk of COVID is much higher than the risk that the vaccine might pose. However, if the pregnant woman is always wearing a mask, social distancing, is younger, does not have any other comorbidities, can she wait? That's a choice that she will have to make with her family, with her physician, and with her obstetrician. The other thing pregnant women can consider is if they feel that there is a risk to the fetus, which so far we haven't seen, but there are no studies as yet. So we have to acknowledge that. Until such data is available, pregnant women can choose to wait until after the first trimester to get the vaccine, when the preliminary development of the embryo and fetus has taken place, and any supposed risk can be further minimized. How about lactation? Once again, the vaccine is considered to be safe during lactation. Lactating mothers were not included in the studies of COVID vaccine. The risk of transmission of the vaccine components to the baby through breast milk is considered to be extremely small or none. But the advantage is that if the mother who is lactating gets the vaccine, when she mounts an antibody response, some of it will be transferred to the baby through breast milk, and therefore the child will have passive immunity for some time. So even lactating mothers, the general guidance is that the vaccine should be safe, but more data are yet to come. Will COVID-19 vaccines cause infertility? The short answer is no, it does not. Who must delay getting the vaccine? So if you have, when should you delay getting the vaccine? Now, one reason is if you're in quarantine from a recent exposure to COVID, then you have to wait until you're out of quarantine. If you've had COVID, you've tested positive for COVID, then it is best for you to wait anywhere from 6 weeks to 3 months after you've had COVID. And the reason for that is because the infection itself mounts enough antibodies in your system that the chance of you getting COVID after a previous episode is extremely uncommon in the 90 days after. So you can delay the vaccine and extend the duration of protection beyond those 90 days. Or if you have received convalescent plasma or monoclonal antibodies, then you also have to wait for 90 days. So the other question that's frequently asked is, can someone who's vaccinated still transmit COVID-19 virus? And this goes into the whole discussion of, can we stop wearing masks and social distance after we get the vaccine, right? So it's not yet known, it's not been proven as yet that once you get the vaccine, you cannot be infected. Meaning, if you've had the vaccine, it's beyond 2 weeks after you've had the vaccine, there's a chance that you can still be infected with the virus. You may carry it in your nasopharynx. You may be asymptomatic, but you might give it to others who have not been immunized or who are immune suppressed. So that is currently being studied by the NIH. The data is yet to come. The risk is there, it is minimal. We don't have data as yet. Therefore, it was based on this that CDC said in the last few weeks that those who have had the vaccine and it's been more than 2 weeks, they can probably congregate in small groups, even indoors without a mask. However, if it is in a larger group, there are other people without vaccine efficacy, or if they have not had the vaccine, or if there are immune suppressed or elderly who haven't had the vaccine, then the risk still probably exists until we know more. Will vaccines still work against emerging strains of COVID-19? I think we already addressed that. There will be some protection, but sooner we all get the vaccine, we can hopefully prevent these viruses from coming in. Now, this is another question. How long can the second dose of COVID-19 vaccine be delayed for those vaccines that need to be given as 2 doses? So both Pfizer and Moderna, the studies were either 21 days for Pfizer and 28 days for the Moderna vaccine. And if possible, the second dose should be administered as close to the recommended interval as possible. But if for any reason, let's say you get sick, you cannot get the second vaccine, there are no appointments, etc., if the second dose of the vaccine is delayed, they can be administered up to 6 weeks after the first dose. There is no loss of immunity, but you can still get the vaccine up to 6 weeks. And there is limited data for other vaccines. Now, on similar lines, the other question that has come up is, how about just 1 dose of the vaccine? Does 1 dose work just as well, or does it not? If you look at the efficacy data after 1 dose of the vaccine for the mRNA vaccines, 2 doses clearly were better than 1 dose. The times when 1 dose can be sufficient is in people who have previously had COVID. There are several papers recently published within the last 2 weeks where data is emerging that someone who has had COVID before, if they get a single dose of either of the mRNA vaccines, the amount of antibodies that are produced in their system is extremely high after a single dose, and it is even higher than 2 doses of the vaccine that are given to people who have not had COVID. So perhaps in people who have had COVID, 1 dose might be sufficient. But what we don't know as yet is how long does a single dose immunity last? Is it better to still get 2 doses? That data is yet to come. When can I travel internationally after COVID vaccine? Are there any restrictions? Maximum immunity to any of the vaccines is after you complete 2 doses, at least 2 weeks after that. If it's a single dose vaccine like J&J, about 2 weeks after the single dose. If you're traveling, it's important to look at local requirements, look on outbreaks and variant strains in the country or a state that you're planning to travel, and see whether vaccines have been implemented in that country or region. And you also need to determine the extent of vaccination in the population that you're going to be exposed to, and continue to review travel restrictions because they change all the time. And just because you're vaccinated, if I were you, I would not let my guard down and not wear masks or socially distance when you travel, because as I have demonstrated before, there are several variants of the vaccine. We may know of some, there may be others that are out there that have not been genetically examined as yet, and they might be much more resistant than the ones that we know. So if you're traveling, especially internationally, and if you have to travel, continue to take precautions. And it might be a great idea for you after you come back to get tested for COVID to make sure that you were not infected when you are away from the country. Should we delay getting COVID vaccine if you've had another vaccine recently? So answer is probably yes. So if you should delay your COVID vaccine if you've had another vaccine within the last 2 weeks. And if you had COVID vaccine first and you need a different vaccine, probably wait a couple of weeks to get another vaccine. This is more to make sure that the side effects don't overlap, since COVID vaccine is new. And also, we don't know exactly how the various vaccines will interact. We give multiple vaccines at the same time, those that have been out there for a number of years. But with COVID, it is best for you to separate the vaccine dosing. Is it okay to schedule a routine mammogram if I've received the COVID vaccine? Answer is yes. If it's time for a mammogram, please get it. And the reason for this question is because if you've had the COVID vaccine, then you may have lymph nodes in your axilla that might be enlarged. And this might show up on your mammogram or any imaging that you have for routine breast cancer screening. And you may have to consider that as a potential possibility, because the first thing that the radiologist might wonder is if you have cancer, and that's the reason why the lymph nodes are enlarged. So you have to bear that in mind. Is it safe to take pain and fever reducing medications like acetaminophen or ibuprofen before or after being vaccinated with COVID-19? I would say don't take these medications preemptively or before you get the vaccine, because you may never develop the fever. It might suppress some of your other reactions. If you get a fever, if you get chills or rigors, yes, please go ahead and take preferably Tylenol, because the Motrin and other drugs in that class, the non-steroidal anti-inflammatory medications, might in some ways suppress a portion of the immune response. It's not been proven, but I would stick with Tylenol. If I'm scheduled for surgery, should I delay my COVID-19 vaccine? Answer is it's probably a good idea to wait until after you've had surgery, because if you get the vaccine right before surgery, 5 to 10% can have a fever. So if you have a fever, then your surgery will be postponed, especially if the surgery needed to be done. If you don't get a fever before surgery, after the vaccine, But if you get a fever 2 to 3 days after surgery, then the surgeon might be concerned that you may have had an infection after the surgery, a postoperative complication. So it might be better to either wait until after surgery, or if it's an elective surgery, if you want to delay, that's your choice. Egg allergy and COVID vaccine, no contraindications. None of the vaccines are made with eggs. If you're getting a antibiotic, should you wait to get the vaccine? Answer is no, because antibiotics don't have any effect on viruses. So those are the questions that I had. I know it's about 8:06. Mr. Pavan, do we have any other pressing questions that I have not addressed?
Yes, Doctor, we have a few questions, and I know we are over the time, so I'm hoping that you will be able to give us another 10 minutes of your time. And
oh, absolutely. I'm here, so
Thanks.
if there are more questions, I'm happy to answer them. As long as our audience is participating, I'm happy to be here.
Sure. Yeah. So we are seeing a few questions coming in. I think one of the questions was around travel. How long before we can travel, and I think you already covered that. So depending upon where we are going, take precautions and study the area and the region where we are going and take precautions. I think that was one of the questions that was asked. There was a question around the mRNA vaccines in particular. The technology has been around for some time, but why is it that it is being used now? And mRNA vaccines, in some cases, have been known to lead to autoimmune diseases. Is that something that we should be worried about?
mRNA vaccines have been studied over the years, but this is the first time that they have been used in clinical practice. Is there any long-term consequences of mRNA vaccines for someone to develop autoimmune diseases based on the mechanism of action of mRNA, how quickly the mRNA is degraded, and what is left is the spike protein itself, which will stay for several days or weeks, and then we develop antibodies to it, and the rest of the vaccine components are completely gone from our system. Therefore, my feeling is that the risk of developing autoimmune diseases as a result of mRNA vaccines should be extremely small or none. But once again, the vaccine is a year old. Whether this vaccine or other vaccines will have long-term consequences, time will tell. But the data is very suggestive. And even though the mechanism and the technology is new, it's been studied for years. And the biggest advantage of mRNA vaccines is how quickly the vaccines can be produced, as we have demonstrated, number one. And number two, how quickly the mRNA can be altered if the virus changes to make new vaccines that can be effective against variant strains.
Thank you, Doctor. There is a question around the India vaccines, and we have received many, many similar questions around this. Is what do you recommend, Covaxin versus Covishield? Is one better than the other?
For Covishield, which is the adenovirus vaccine, we have more published data. We have data from their trials that were done in South Africa, UK, and in Brazil, as well as the data that was recently released from their studies here in the United States that was released last week. Covaxin also has data. What they have done is they have published their phase 1 and phase 2 data earlier in March, and their phase 3 data, there was a press release a few weeks ago by the company, which indicated that it was about 80% effective. So I would not have any reservations with regards to getting either of the two vaccines. It is just that for Covaxin, we need to see published peer-reviewed data, which is hopefully coming soon.
Thank you, Doctor. So one question around, is the current precautions that people have been taking in terms of, getting groceries and things like that. Do those still have to be wiped down, or do we still need to continue to follow the precautions post-vaccination as well?
That's a tough question to answer. One is, it depends on the level of virus that's circulating in the community that you are living in. Meaning that if there is a significant proportion of people in the area, in the town or city that you're living, or state has had the vaccine, and the local numbers and trends are going down, then it is left to your discretion as to when you might want to cut down those measures. In general, I would use common sense with regards to how meticulous you need to be when you buy groceries and then wipe it down. It all depends on your comfort level and what you would like to do. But keeping precautions, more than that, I think it's the social distancing and wearing masks until such time that the community level of the vaccine goes down and a significant proportion of the community has been vaccinated. I would like to stress that more, and not to let the guard down before a significant portion of the population has been vaccinated.
Thank you, Doctor. One question is, states like Arizona have opened up, public places now, and even before 80% of the population got vaccinated. What is your suggestion around, publicly, mingling, etc.
Well, the answer is similar to my previous answer, which is each citizen and individual will have to make his or her choice. I would like to be educated on what the local rates are and local vaccination rates are, and I would use that as a gauge to let my guard down. Not because the state changed requirements. I would say the vaccine was open to those over the age of 16 recently. Until such time that a majority of the population gets the vaccine, 2 doses of the vaccine, and they're immune to it, it is probably best for us to stay safe under the circumstances.
Thanks, Doctor. Last couple of questions. Why is the side effect more prominent in lesser than 55 age group? And why are variants seen or originate only in certain countries and states?
The reason why side effects of the vaccine is more common in less than 55 is because the immune systems of those who are younger tend to react more strongly to external influences, including antigens and infections, compared to those who are older. That's a general statement, but that's the main reason why side effects can be more common. And once again, common side effects are still they're infrequent and they're minor. Even if someone has fevers or chills, it lasts for a day or two and it goes away. Happened to me, and after about 2 days, I was fine. The second question was, why are variants more common in some countries? I don't think it is that the variants are more common in some countries. It is that some countries are testing for the variants more often or more frequently than other countries, because some countries have a national database where the viruses, thousands of viruses are being tested, and United States was actually lagging behind some of the other countries, in fact, and we have really picked up pace in the last few weeks about testing more of these strains for variants. But sometimes it is just a matter of geography, matter of population density, and how often infection stays. So it's hard to predict as to where the virus might mutate next, because it has happened in various geographic areas, going from between South Africa to London to here, and then in California, you're seeing some of the similar variants here too.
Thank you, Doctor. That was last of our questions. Any closing comments, any final guidance that you might want to provide to the community?
No, I think I really enjoyed this session. I hope those of you who joined our session on a Saturday evening got something out of this session. I would like to sincerely thank each and every one of you for joining the session. If you have any other questions, please feel free to reach out to me. And I would also like to thank Kannada Sangha of Arizona for doing such a great job of arranging this meeting and for conducting the session today. Thank you very much.
Thank you, Doctor. I don't have words enough to thank you for actually taking the time on a Saturday to come and educate us and the whole community about the vaccinations. We are over time as well, so thank you for that as well. We do thank the India Association of Phoenix and the Lions Club in partnering with us in bringing this session so that we could reach out to a wider audience and wider community. Overall, it was an excellent session. Thank you for so much of information that you have provided. I think it definitely helps, and it helps the whole community. Thank you so much for your time, Doctor.
Thank you again, and have a great evening. Bye now.
Thank you. Bye.